Yeah, so I'm afraid it's going to be a little bit of a lecture, but I'll try to keep it brief and clear. Yeah, so before I start on our own research, there are essentially two types of stem cells, and they often get confused. There are the embryonic stem cells that only live briefly directly after fertilization, and they build an entire body. So these stem cells can make everything. And the embryonic stem cell, of course, has lots of ethical and logistical problems, but there is a synthetic version of the embryonic stem cell, which is called the iPSL, which can be made from essentially any human cell type. And it has the same capacity of being able to create any part of the human body. So that's the so-called pluripotent stem cell. So that's not what we work with. We work with adult tissue stem cells. And of these, there are many. The best known was discovered 6-7 decades ago, the bone marrow stem cell, which can make all the blood cells lifelong. We probably, I would estimate, in our body have at least 100 different adult stem cell types. And what they, in essence, do is they maintain our bodies lifelong. So cells work hard, they get lost, they have to be replaced. Stem cells do this, so every organ will have its own dedicated stem cells. When they sense cells are lost, they basically divide a few times to replenish, to replace the cells that are lost. So there will always be stem cells in your organs, and those stem cells will constantly make additional cells to fill the ranks of the cells of the tissue in which they live. So I already mentioned the bone marrow stem cell. When we, through serendipity, basically by chance, discovered that molecules we were working with were key to mediate so-called Wnt signals. And Wnt signals were known to be important during embryonic development, when cells rapidly have to take all sorts of different decisions to finally build an entire body. There's a handful of signaling pathways that mediate this. They're called Hedgehog and Notch and Wnt and a few others. And we found a key component of the Wnt pathway while we were essentially studying T lymphocytes. We're not looking at all for this. But this actually, we realized this is interesting. We then started studying the Wnt pathway, not in early embryos, but in postnatal mice. And we realized that Wnt factors secreted peptides, secreted proteins, not only control early development, but after birth, they control many, if not all, of the stem cells that maintain, these adult stem cells that maintain our bodies lifelong. And we found this because when we knocked out this unique component of the Wnt pathway that we discovered, these mice get born, but then they die in about 5 to 6 days because they fail to maintain the inner lining of the gut. This already put us on the track of Wnt signaling is somehow related to the activity of stem cells in the gut. That was what we showed, but we now know probably in all of our other organs. At the same time, we discovered that mutations in the Wnt pathway, this was done together with Bert Vogelstein at Johns Hopkins, that activating mutations in that pathway are essentially by far the most important cause of colon cancer. So on the one hand, and you have to realize we were immunologists. We never thought about the gut. I knew where the gut was from my medical school, but that was about it. But we realized that the same pathway maintains the inner lining of the gut lifelong and actually takes care that gets replaced every 5 to 7 days. So it's by far the most rapidly self-renewing tissue of our body, all driven by these Wnt signals. And then the cancers of that organ, colon cancer, is almost always driven by activating mutations in that same pathway. So we realized the biology of stem cells of the gut and the biology of cancer are very, very similar. They exploit the same principle of Wnt signaling. Now, it then took us a number of years. I'm now talking about the mid-90s. I converted my lab into a gut biology lab and actually moved it to another institute. I used to be an immunology professor. Now I'm a professor in molecular genetics, and I then became director of a developmental biology institute, a basic science institute. And so there we eventually discovered these enigmatic gut stem cells. This was done by Nick Barker, who is now in Singapore, British young postdoc, who found a marker, LGR5, that revealed the presence of tiny cells in the lining of the gut that were constantly dividing And when we marked them genetically, we could show that they would make rapidly every other cell type of the same lining of the gut. So within 5 days, if you mark, at least in mice, if you mark these putative stem cells with a particular color, 5 days later, the entire insides of the gut have that color. And that implies that these cells constantly replace all other cells in the gut. There was almost a mathematical proof of that LGR5 marks these unique stem cells. One line of research there was that we showed that LGR5 does the same thing in probably all other internal organs, although those stem cells will not make gut, but they make liver or they make lung, but they're always marked by LGR5. And we started learning about how these stem cells actually work in the gut. And that was very important because there were all sorts of dogmas around stem cells. They're magical, they're very rare, they are very small, difficult to find. They don't divide, or they divide very rarely, because if a stem cell would divide, it has to copy its DNA, could make mistakes, and maybe become a cancer cell, or maybe die, and then you would have an organ with no stem cells. That would be pretty bad. So the idea was stem cells can be defined as cells that rarely divide. There was, in all the textbooks, every stem cell lab in the world looking for stem cells, they were always looking for cells that were not rare, that were rarely dividing. Now these gut stem cells divided every day. So in the lifetime of a mouse, one stem cell will divide 1,000 consecutive times, and for us, for humans, probably 20,000. Now, when we noted that these stem cells were essentially not quiescent, and that's the scientific term, but they were constantly dividing. First of all, we had to fight quite a bit to convince the field that these were stem cells, because everybody knew that stem cells rarely divide. So this took actually 4 or 5 years to actually convince the entire field that this is the real thing. These are the stem cells. Meanwhile, Toshi Sato, a Japanese gastroenterologist in my lab, and Nick discovered the stem cells 2007. We noticed how they were behaving. They're dividing constantly. Toshi Sato said, when he entered my lab, he said, Hans, we will try to grow these stem cells because they divide so rapidly in a mouse and in a human gut. It should be possible to rebuild that environment in a plastic dish and have them play the same trick. And to be honest, our intention was really to take one stem cell and turn them into 1,000 or 1,000,000 stem cells as a lump of stem cells. Now, I had tried to convince other people in my lab to try this experiment, but there was another dogma. So the first dogma is stem cells don't divide. The second dogma was normal tissue cannot be grown, cannot be expanded outside the mouse or human body. Normal cells will maybe stay around for a week or 2 weeks, but they will die. And it's only cancer cells that will continue to grow. And it's true, every cell line in the world that people have been using for research or for drug development, these cells are cancer cells. If you transplant these cells into mice, they will make tumors. They will not make normal tissue. And so people in my lab said, no, I'm not going to waste my 3 or 4 years in your lab on a project that's impossible. But Toshi actually had tried it in Tokyo earlier. We sat down and we figured out, what are the growth factors that stem cells need, that they use in the crypt to be so active, divide every day, lifelong. And there were only 3 that we could think of. And then we'd heard from Mina Bissell that Matrigel is a magical substance. It's a gel at 37 degrees. It's a liquid at 0 degrees. So you can put the cells in on ice. Then you warm it up. It becomes a little pudding with the cells in. It consists of collagens and laminins and other extracellular matrix components. So exactly the environment that the stem cell should like. And then just adding these 3 growth factors was an immediate success. So a single stem cell in this gel with the 3 growth factors started dividing. And that was, of course, what we hoped and what everybody said was impossible. But then we realized that they were not just making stem cells, they were making structures. And this was, I think, the biggest surprise, that first of all, they never stopped growing, but rather than just replicating themselves, they produced more. They produced what Toshi immediately called mini guts. And indeed, they were almost perfect copies of the inner lining of the small intestine or of the colon. You have to realize there's about 10 cell types that we use to digest food very efficiently, to take up the nutrients, to produce mucus, all the other hormones that are produced in the gut, the protection against the microbes is all done by these 10 cell types. Now, this one stem cell in this very simple culture medium in 3D, in a gel, will actually produce a structure that has all of the other cell types in the right numbers, in the right places, and it can do so for many, many years. And we first thought that they were growing so fast, something must have gone wrong. They must have turned into cancers. We then did a transplantation experiment with Mamoru Watanabe in Tokyo, and that experiment showed that you can take a single stem cell, and I think it's really remarkable, grow it up to maybe 1 billion cells in the form of these mini guts, transplant them to, in this case, the first experiment was mouse to mouse. Actually, currently he is doing human to human. You can actually then transplant them to mice that have inflammatory bowel disease, and these small mini guts, totally synthetically grown from one stem cell, you can make millions out of them. They actually, when you bring them into the lumen of the colon with inflammatory bowel disease, they find the lesions, and like a living Band-Aid, they will land on top of these lesions and they will fill up the gaps and then persist as normal cells for the rest of the lifetime of a mouse. So really remarkable that a single stem cell in a very simple environment will grow forever and will make normal gut forever. And even when you transplant it back, it makes normal gut and it actually cures these mice. And then maybe a last sentence. Once we knew how to do this, we then slowly turned to many other organs. Already said that these organs will also have stem cells that have the same LGR5 marker when they're active, but they have different growth factor requirements. So you need to adapt a cocktail of growth factors for every new tissue. You have to think of liver, lung, prostate, breast, pancreas, etc., etc. But with some tinkering around, we usually can come up with a cocktail that allows the expansion of little bits of tissue in the form of these organoids, mini lung, mini liver, for years. And then, but we'll talk about it later, I guess, we realized that this was quite incredible technology, because almost any biological experiment you would do in animals or with cell lines, you could actually do better in these human mini organs or organoids. I think I'll stop here.